Okay, I'm going to talk about mycorrhizae, which are plant roots and fungi today, and I want to acknowledge that all the video that I've got, I came from the internet and from the literature. Major objectives that I'm going to try to address during the talk is to introduce the major types of mycorrhizae and examine the types of plants and fungi that are involved in each one, and then describe the interactions that occur. Otherwise, who gets what and the interactions. And then briefly toward the end, discuss some of their importance in ecology. Mycorrhizae are widespread interactions between fungi and plant roots, and they're primarily focused on vascular plants. So we're talking about angiosperms, gymnosperms, ferns, and even some mosses, which aren't vascular, have mycorrhizal association. And that appears to be the norm for most of the plants. Somewhere around 90 to 95% of plants can form mycorrhizae, and some 6,000 species of fungi. The interactions can vary depending upon the organisms involved. In most cases, they're mutualistic, in that both the fungus and the plant benefit. In some instances, the fungus may parasitize the plant, and the plant, as we'll see toward the end, can parasitize the fungus. In order to get started, I need a few terms to make sure that everybody's on the same page. Basic unit of a fungus is called a hypha, plural being hyphae. Basically what that is a tube of cytoplasm surrounded by a cell wall, which you can see in this lower figure. The mycelium would be all the hyphae in a particular fungus body. So the fungal body would be called a mycelium. And hyphae can either be septate, in which the hyphae have cross walls, as indicated in this figure that's stained in purple, where you can see the cross walls in the hyphae. Or they can be aseptate, in which the hyphae don't have the cross walls, as indicated in the picture on the right. There you see the hyphae, but there are no cross walls, so they're referred to as aseptate. Septate hyphae are typically characteristic of the higher fungi, the ascomycota and the basidiomycota, and aseptate hyphae are found in the lower fungi. Types of mycorrhizae that we'll briefly introduce are indicated as either ectomycorrhizae, sometimes called sheathing or ectotrophic mycorrhizae, in which the hyphae of the fungus don't penetrate the plant cells. So they can intertwine among the plant cells, but they generally they do not penetrate into the cell itself. And then an endomycorrhizae, we have where the fungal hyphae penetrate into the cells of the root. And we can divide those into aseptate and septate mycorrhizae. The aseptate are probably the most widespread types of mycorrhizae that one finds, and they're referred to as the arbuscular mycorrhizae, or abbreviated AEM. And there are a number of septate hyphae that are endomycorrhizae. Plants in the Aracales can include mycorrhizae that are ericoid, or butoid, or monotropoid. So there's basically three types of mycorrhizae that have septate hyphae in the Aracales. And then the orchidaceous mycorrhizae are septate hyphae that are associated with orchids. We'll talk about those briefly. Ectomycorrhizae are the most conspicuous and easily recognized of all the mycorrhizae because the plant roots are enclosed by a sheath of fungal hyphae. So the fungal hyphae form a sheath around and modify the plant roots so that they can be recognized externally. The mycelium penetrates in between the cells of the cortex, but it doesn't actually penetrate into the cells themselves. So the fungal mycelium is intimately associated, as we'll see in a couple of minutes, with the cortex of the root. And the fungal tissue can account for up to 40% of the mass of the root. The hyphae also extend out into the soil, and we'll refer to those as extra radical hyphae. In this little diagram, you can see mushrooms with their hyphae coming down, and the mycelium permeates throughout the soil and then can form mycorrhizae with plant roots. In this case, a tree. They may go over here and associate with this particular tree as well. And there you have some puffballs that form mycorrhizae and have their mycelium extra radically that extends out into the soil so they can useful in absorbing nutrients from the soil. So if we look at a cross section of a root, then we can see that you have this sheath of fungal hyphae around the outside of the root, and then the hyphae may extend in and go between the cells of the cortex. They never get, although they can go between the cells of the cortex, they never get past the endodermis, so they don't get into the vascular bundle. Characteristic of this is the Hartig net, and that's the hyphae that form between the cells of the cortex, and that's diagnostic to see if these are, if the shape of the root indicates a true ectomycorrhizae. They have to have a Hartig net if they're not. And this is a micrograph of the cortex of the root with the Hartig net, the hyphae in between the cortical cells. The absorbing roots are those that are affected, and they become more thick and branched repeatedly after they're infected. In the figure on the right, you have pine roots that are not infected with mycorrhizae, and you can see the branching pattern on those. And on the left, you see the pine roots that have mycorrhizae formed on them. And they have much more branching, they're much thicker, and they don't have root hairs because the mycelium, the hyphae of the mycorrhizal fungus, is what allows them to absorb. So they become thicker and more repeatedly branched once they get to mycorrhizae. And here are some better pictures of mycorrhizae. And you can see these are all micro. So if you dig up a plant, and I used to, when I lived in Alabama, I had a bunch of little pine seedlings growing in my backyard, and every time I'd teach this, I'd go up and dig a little seedling up, and it'd always have mycorrhizae, so I could take them in and demonstrate them. And then you have the extra radical mycelium that extends out into the soil, and that's going to be real important when you talk about what these things do. So those are what you'd see if you dug up a pine tree or some other kind of a tree that has these ectomycorrhizae. What are the symbionts for the ectomycorrhizae? Well, as far as the plant species are concerned, we're mainly talking about woody species. Trees, shrubs, eucalyptus, all can form ectomycorrhizae. Tropical trees typically don't. So we have the major species of coniferous and deciduous trees that form ectomycorrhizae, and it's rare to go out in the woods and find uninfected trees. In some trees, the association is obligate. In other words, only a particular fungus can form an association with the tree. In other cases, it's facultative, where they may be or not be. Since trees are important in forestry, then you can see that these mycorrhizal associations are going to be important in forestry situations. As far as the fungal symbionts that occur in these ectomycorrhizae, you have mainly basidiomycetes, lots of mushroom species, puffballs, and related species. I got one down here because I just love to say the name Piscilithus tinctoris. It's used to form commercial inoculum for nursery trees, and it's real common in the southern, southeastern United States. They inoculate their trees. Forestry people inoculate trees with this one, and this is a picture of it. It's kind of an ugly-looking puffball kind of a thing. It forms literally gobs and gobs of spores. But that's a common one that they use to inoculate trees in the southeast. Also, some ascomycota, cup fungi, and truffles and truffle-related organisms will also form mycorrhizae. And this is a picture of a truffle. I couldn't get a real truffle to show you because they're too expensive. And there's about 5,000 species of fungi. Typically, for fungi that form subterranean fruiting bodies like this truffle, and there's a lot of other species that just aren't edible or not tasty like a truffle, about all of those are mycorrhizal, both ascomycota and basidiomycota. will produce subterranean some subterranean fruiting bodies. So what's the specificity of the association? There's a lot of variability. And most tree species will form mycorrhizal associations with a lot of different fungal species. So you may have different mycorrhizal fungi on the roots of one plant. Some fungi are fairly specific, and then they'll only form associations with one plant species. And what you do is you find that these mushrooms are going to be found in stands of that particular tree. So if you're looking for a particular mushroom that's specific, then you want to find a stand of those trees to go and look for those particular mushrooms. Others are not very specific, so they'll be formed on a lot of them. Up in the Northeast, they've studied these a lot, and Douglas fir has been studied very, very widely, and they found something like 2,000 species of fungi identified from its roots. So they can form associations with lots of different fungi. And on the roots of one plant, one tree, you might find lots of different fungal species. If you go out in the forest, then a high percentage of the mushrooms that you find are going to be mycorrhizal. This is a table that was done in a Pacific fir forest. And this means above ground fruiting bodies like mushrooms or puffballs. They found 28 species, and 71% of those species were mycorrhizal. So the majority of the mushrooms that were found in this particular forest were mycorrhizal. The one species that was a below ground fruiting body was also mycorrhizal, so it was 100% in that case. So most of the mushrooms that come up in the forest are going to be from mycorrhizal fungi. How does one detect these if they're down in the ground and they're covered up by soil and they're surrounded by the roots? Well, one way that people have done it is to go out and count and identify the fruiting bodies. So if you see these fruiting bodies and you can go to the literature and see if they're mycorrhizal, then you'll know that what might be there. People also take soil cores and then they separate and identify the mycorrhizal roots by their morphology. And of course, you got to determine if they have a Harding net to make sure that it's a true ectomycorrhizal fungus. And now people are using molecular methods. And that way you don't have to wait until mushrooms come up, but you can go out and sample the roots and extract DNA or RNA from the roots and then do molecular methods to try to determine what species of fungi are present there. Okay, what's the characteristics of these ectomycorrhizal fungi? Most can grow saprotrophically, so they don't have to be associated with the tree. And a lot of them have been cultured. They can grow in petri dishes on artificial media. So they're not totally tied to the tree. But most of the ones that have been studied like this, cultured and grown, don't degrade the complex plant polymers that one sees out in the environment. So they're not good decomposers. They can't degrade cellulose and lignin and other kinds of complex polysaccharides that you find in plant litter. So they basically depend upon soluble carbohydrates, sugars like sucrose that they get from the plant. They can also have various organic growth factors. They may need vitamins or amino acids that they can get from the plant. So these fungi are generally not decomposers, but they're dependent upon the plant for carbon and energy in the environment. And they get their sugars from the plant, from the plant's photosynthate. So what are the benefits to the fungus? I think that's pretty obvious. Whenever you have a heterotroph and an autotroph coming together and making them a symbiotic association, the heterotroph is going to get its source of carbon and energy from the autotroph, because plants carry out photosynthesis and have an abundance of sugars and other kinds of carbohydrates that they can provide to the fungus. And it's been shown that if you provide plants with labeled carbon dioxide that's taken up and made into sugars via photosynthesis, that radioactive carbon then is going to appear in the fungus. The sucrose, which is the common disaccharide that plants make, once it gets into the fungus, it's converted into another disaccharide, trehalose, or a sugar alcohol, mannitol, by the fungus. And the fungus does this because those things can't go back into the plant. So the plant can't get those things back once it gets taken up by the fungus. And I've seen various and sundry estimates wherever I read something that anywhere from 10 to up to 30% of the photosynthate produced by trees is passed to the mycorrhizal fungi. That's a big percentage. If 30% of the photosynthate produced by trees goes to the fungus, then you would think that the tree wouldn't grow so good when it's got these mycorrhizal fungi on it. But there's been a lot of studies that have shown that tree growth is actually much better when mycorrhizae are present. And as you can guess, which one of these has mycorrhizae in it? This one over here on the right. The left is grown in soil without mycorrhizae. These are Monterey pine seedlings grown in a prairie soil, non-mycorrhizal, mycorrhizal, and you can see the growth is much better, even though a lot of that photosynthate that's being produced by the plant is going down into those fungi. And if you don't like pictures and you'd rather look at numbers, here is a growth of Sitka spruce with a basidiomycete in culture in greenhouse, and basically it grew for 14 weeks. And if you look at the total biomass in the control, which has no mycorrhizal fungus with it, versus the biomass of the plant that has the mycorrhizal fungus, you can see it's more than double its biomass. And if you look at the number of root tips, that's like what? That's like 4 times as many root tips in the mycorrhizal seedling than in the control. So growth is better typically with the mycorrhizae associated with the roots. So basically, when you go out in the field and you're looking at a plant, you're really not looking at a plant most of the time. You're looking at a plant associated with a mycorrhizae. So what can the benefits to the trees, if they grow better, what can the benefits to the trees be? Well, fungi are going to increase the supply of nutrients that are typically limiting to the tree. And these are going to include particularly phosphorus, but also nitrogen and water. Phosphorus is generally present as phosphate, and that's insoluble in most soils. So it's not going to be moving around. It's going to be stuck in a place. And consequently, for a plant to be able to take up phosphate, it's going to have to get to that area where the phosphate is located to take it up. And basically then what the fungus does is this extra radical hyphae, the hyphae that's out in the soil, extend over a larger volume of soil than the roots do. And that's going to increase its ability to absorb such insoluble nutrients like phosphate. With nitrogen, the nitrate in the soil is more mobile, and so consequently that's not probably as important, because if a plant is taking up nitrate, then it may be replaced by soluble nitrate moving around in this situation. And to demonstrate this, I found some pictures like this of a seedling. Here's a seedling growing in something that's kind of like, does everybody know what an ant farm is? Where when you were a kid, you had an ant farm that had a glass on one side and kept it dark. And then you could take the thing off and you could see where the ants went. Well, that's basically what they did here. They had a glass on the side of a soil, and they planted this seedling, and they also put some fungus in there. And the root is probably maybe a little bit bigger than the seedling, so it maybe goes down about that much. And all the rest of this is mycelium from the fungus, the mycorrhizal fungus. And you can see how it explores a much larger volume of soil than the plant seedling can by itself. So it could then maybe take up phosphorus that's over here that the plant wouldn't be getting. And that would then increase the plant growth, because sugars generally aren't going to be limiting to a plant that's living in the sun. Now here is another example of that kind of a thing, except here what they did, they had several seedlings. And before they planted them, they put in radioactive phosphate. So radioactive phosphate has been distributed in the soil in this particular little situation. And then they let the mycorrhizae and the roots grow in. And then after they'd grown for some period of time, they did what's called an audio-radiogram, in which they put a film emulsion over the surface, and radioactivity will expose film so that when they develop the film, they can see where their phosphate is, where the radioactivity is. And that's indicated in this picture on the right. And there you can see that where they put an arrow here to show particular mycelium of this fungus. And over here, you can see it's really accumulated the phosphate in that particular area. And you can see a lot of those areas around here where phosphate has been accumulated by the fungus and then taken up and put it into the plant tissue. So this, if phosphate was limiting in a particular environment, then this would allow for the plant to accumulate more phosphorus, and it could grow better and grow faster. So fungal mycelium in the soil increases the nutrient uptake. If you look at this little diagram, you have the plant root growing in a soil, and it will have a certain depletion zone where the roots could take the nutrients up. But if you have mycorrhizal associations that form a larger volume of soil being explored, then you can look at nutrients being taken up in this area as well. So the fungal mycelium then increases the surface area and volume that the plant can absorb nutrients from. In addition, nutrients that are present, but they may not be available for the plant because the plant can't digest it, or it may not be able to solubilize that nutrient, whereas the fungus might be able to, then those nutrients would also be available to the plant. So the fungus may be able to digest particular compounds that the plant couldn't take up, and that would make those nutrients available to the plant. And then fungi can excrete chelating agents that solubilize nutrients that would allow for them to be solubilized and then taken up by the plant. Okay, some other benefits to trees. Fungi can produce plant hormones, and that changes the physiological state of the root so that the root is more physiologically active for a longer period of time, and that increases nutrient and water absorption. There have also been studies to show that tolerance of plant to drought, to high temperatures, pH extremes, and heavy metals is increased when they're mycorrhizal. So in that case, if one's planting, for example, trees in an area like a mine, reclaimed mine spoils, then you may have pH extremes or heavy metals that would be more overcome by seedlings if they had mycorrhizae already on them, because the fungi give them some tolerance to that. And it also, as you might imagine, increases resistance to infection by root pathogens, because you've got this sheath that provides a physical barrier and it prevents other fungi from coming in and colonizing the root. So ectomycorrhizae are definitely mutualistic symbiosis. Both the organisms benefit from that association. And as I've mentioned, seedlings in nurseries can be inoculated with the fungi so that when they're planted, they'll have a better chance of success. And this little diagram shows a mushroom with mycorrhizal associations. And here's a subterranean fruiting body that's made in mycorrhizal associations. And then you have this cute little rodent trying to dig up and eat this sucker so it can spread the spores around and colonize other trees. Okay, I think we're going to stop here and see if there's any questions. Are there any? I don't have anything that I can see. Anybody have any questions on the ectomycorrhizae? No? You're muted.
Yeah, I have a question, Keller.
Okay.
So can you buy different mycorrhizae? For example, if I want to have a successful transplant of manzanita.
Of what?
Can I
Manzanita?
Manzanita.
Then I would try to get soil from around the manzanita that you're getting it from.
And can I supplement that with something that's on the market? that I could buy and sprinkle like fairy dust around it?
You can buy mycorrhizae, but you don't know. I mean, the manzanita is going to have a specialized, it's not going to be just any fungus that you can throw in there.
Okay. And so then
So,
there were ratios, say, from the size of the plant that you dig up to transplant. Is
yeah,
there some kind of a guidance as to how much soil around it you should try to get?
as much as you can, and then I'd take some extra, just in
Okay.
the, you know.
Okay.
And
Thanks.
I'd get some more. When I went out and tried to get some of my manzanita, I took a tub and filled it up with the soil around it.
Oh, okay.
And
All
that
right.
I see now. I have.
Thank you.
Sure. Now, you can buy mycorrhizae, particularly arbuscular, and we'll talk about those next. But you never know if you've had success
Oh, okay.
unless you've got a microscope and the procedure to see if they've come in.
Yeah.
With the ectomycorrhizae, you can see them if you dig up the roots and you can see them on there. But with the other ones, you can't tell.
Okay, so that leads me to a related question. You said you can see them. Can I see them with the naked eye, and what would they look like?
Well, for the ectomycorrhizae, you definitely can. I showed you some pictures of those right at the beginning. Did you see
Okay,
those?
with the naked eye, you can see them?
Yeah. Oh, yeah. They're fairly obvious. Let me. How do I? Well, I can go back this way. There. They look like that.
Oh,
And
okay.
And that's, well, I don't have a scale on there, but yeah, those are definitely visible.
Okay. All right.
There's roots that have
Okay, so,
mycorrhizae and roots that don't have mycorrhizae.
all right. Okay, thank you.
Sure.
So
You can
Keller, Jackie Blurton has a question.
Okay.
Go ahead and unmute, Jackie.
Hey, Keller, can you hear me?
Yeah.
Okay, I have a question about how some invasive species might affect the mycelium and
You have what?
how an invasive species might affect the mycelium in the soil, like horehound. So I've been cleaning out this patch of woods that all underneath the junipers, there was really dense clumps of horehound.
Yeah.
And so the whole surface of under the trees was just these massive roots.
Yeah.
Does that affect the mycelium in the soil at all? And how the tree can uses the mycelium?
Well, I have no idea. I would think that, did it go past the drip zone of the tree?
Pardon?
Was how big was this patch of horehound?
Oh, it was just completely covering the ground,
okay.
every square foot.
And
So I've dug up a lot of it. So now the soil is exposed, and but the trees in this particular area that had all this carpet of horehound had lots of dead branches, and they just didn't seem to be real healthy. And I'm wondering if, well, it could be that they aren't getting as much moisture because of the horehound, but I was wondering if it affected the mycelium also.
Oh, it could. What kind of a tree is this?
They're one-seed junipers and alligator junipers and pinon pine.
Oh, okay. Yeah, I'm not really sure how that might affect the. I really don't know, but it's possible that if you got rid of it, that might help.
Yeah, I guess it can't hurt because it was doing a lot of harm. But anyway,
put some grass in there.
Yeah. Well, thank you so much. At least it's going to get more moisture and less competition.
Right. You don't ever see any mushrooms coming up, do you?
I'm going to look.
Okay.
So Keller,
yeah,
I'm just wondering, you're talking about trees, but I'm assuming that there are mycorrhizal associations between herbaceous plants and, things that are not trees. And
but they don't have ectomycorrhizae.
okay, so it's just, okay,
The ectomycorrhizae are specialized on woody plants. You could have shrubs and trees that are woody.
Okay.
But herbaceous won't have ectomycorrhizae.
However, the herbaceous would have endomycorrhizae.
They'll have the next group that we'll have.
Okay, the next group. So what I'm getting at is that there are these associations between the fungi and the trees in one type of mycorrhizae, but there are also associations between, you know, all of these herbaceous and other plants as well. So if we were to look down from you know, with a drone and we were able to see the plants, but then see what was going on underneath, it would just be this massive map, if you will, like a road map, a very busy road map of connections. But then if you dug down and like in a trench, 6 or 8 feet down, and you could look, you know, into the soil, you know, those mycorrhizae are going down into the soil in a 3-dimensional form, and it was just the biomass of that mycorrhizae must far outweigh what we see on the surface.
Oh yeah, but they don't go down that deep. Most mycorrhizal are within the top, I don't know, 6 or 8 centimeters of the soil.
Oh, okay.
Yeah.
Very
that's where the feeder roots typically are. I don't think you see them real deep. The mycelium will be real deep.
Okay.
So, okay. So is there anything else that we will need to talk about with ectomycorrhizae, trees and shrubs that are woody? Then the next group I want to mention are the arbuscular mycorrhizae, and they're abbreviated AM. We don't know as much about these guys as we do about the ectomycorrhizae, and you'll see why in a couple of minutes. But they appear to be the most common type of mycorrhizal association that we find, particularly if you're thinking about the number of plant species that form them. So you find these in all types of terrestrial plants: annuals, perennials, temperate, tropical trees, crop and wild plants. They're estimated to occur on, I've seen 90 to 95% of vascular plant species. The fungi that form arbuscular mycorrhizae are all in the phylum Glomeromycota, which nobody probably that's listening to this has ever heard of, right? Ascomycota and Basidiomycota you have heard of, right? But Glomeromycota is a fairly small phylum of fungi that are aseptate, so they're generally in the lower fungi, but they're right below the Ascomycota. There's only about 200 species here, and they're in 10 genera. And all of them are obligate biotrophs. And there's some terms I didn't talk about, but what that means is they can't be grown by themselves in culture medium. They have to be associated with a plant. They're obligate, so they can't be grown outside of a plant. And they're biotrophs, meaning that they have to get their carbon and energy from a living organism, from a plant. So they're all obligate in that manner. So you can't grow these things by themselves. They do form these very large spores out in the soil, and diameters range anywhere from 50 to 400 micrometers. That's 4/10 of a millimeter. Sorry. So they're almost large enough to see with the naked eye. And this is how you can identify them, the characteristics of these large spores. For the Glomeromycota, the arbuscular mycorrhizae. They found fossils of these spores that are as old as the first land plants, something like 460 million years ago. And there are hypotheses that plants got on land because of their beginning association with these types of fungi. And that might be why we have land plants now, is because these fungi allowed them to adapt to the crummy soils that were there when plants first got out of the water. Well, if we only have, what did I say, 200 species? Is that what I said? Yeah, 200 species of fungi and how many thousands of species of plants, you know there's going to be very low specificity. One fungal species may form associations with a lot of different plant species because there's just not enough fungal species to go around for every plant to have its own fungus. Okay, and here's where the rubber meets the road. The morphology of the root is not modified when these things are there. So you can't pull up a plant and look at its roots and tell if it's got these arbuscular mycorrhizae. To detect them, you've got to clear and stain the feeder roots to observe these fungal structures inside of them. So typically what that involves is you got to take roots out, put them in a solution, autoclave them, and then put some stain on them and then look at them under a microscope to see if there are these fungal structures inside the cell tissues, the fungal tissues, or the root tissues, sorry. The fungi form both intercellular hyphae in between the cells, and they also penetrate into the cells. So they'll have intracellular hyphae. They penetrate into the cells of the plant root. And the intracellular hyphae, the ones inside the cells, no, you don't need to worry about that, but they're called arbuscules, which means that they have a tree-like branching pattern. They're like little trees stuck inside these plant cells. And this is where they think that the nutrient change occurs between the fungus and the plant. Ostoria basically are modified hyphae from plant pathogens that suck the juice out of plants. And there's what some arbuscules look like. There's the hypha, the aseptate hypha, and this is penetrating into a plant cell, and you can see it has little tree-like branches all around. And there are some other ones down here that are the arbuscules. The arbuscules are generally surrounded, or they are surrounded by the plant membrane, so they don't really penetrate into the cytoplasm of the plant. And generally, arbuscules will disintegrate after about 2 weeks in the plant cell, and that's where the nutrient exchange occurs. Stuff from the fungus can be absorbed by the plant, and vice versa, this can absorb stuff from the plant. So that's where nutrient exchange occurs between the fungus and the plant and the root. Many species of the AMs form large swellings called vesicles at the ends of hyphae, or they may be within the hypha. These are vesicles. These are typically rich in lipids, and they're thought to be involved in storage. And when these were first described, most of the fungi, these mycorrhizal fungi had vesicles and arbuscules, and they were originally called vesicular arbuscular mycorrhizae, or VAM. But since then, they've discovered species that don't form vesicles, so they got rid of the V, and they just call them AM now for arbuscular mycorrhizae. But a lot of them will also form these vesicles. And this kind of is a diagram showing you the hyphae that are extra radical that are going out into the soil. They may have vesicles out there. They can form spores out there in the soil. And then they penetrate into the plant and form intercellular hyphae, and the hyphae then may penetrate into the cell and form these arbuscules. And they may have vesicles inside the cells as well. So you can have vesicles, but you got to have arbuscules in order for them to be an arbuscular mycorrhizae. Okay, so the root looks the same. This is all happening in the cortex. That's generally where all mycorrhizae are going to mess around. So as you might imagine, they're not going to be as well characterized as ectomycorrhizae, because you can't go dig up a root and know that it's got a mycorrhizae because it's not altered. You've got to take them and clear them, stain them, and then look at them under the microscope. And you can't grow the fungi, so we don't know much about the fungi because they can't be grown outside the plant. So you can't do a lot of experiments you could do with the ectotrophic mycorrhizae. So what about the interaction? Well, the fungus we know probably receives its organic nutrition from the plant since they're biotrophs. We don't know what their requirements are, but they must have some pretty big requirements if we can't grow them in culture. So they must require a lot of different types of organic materials from the plant. The fungus also produces extra radical hyphae that go out into the soil, and these take up inorganic nutrients from the soil, again, particularly phosphorus. And why is that? Because phosphorus or phosphate is not soluble, and it's going to be localized in the soil. They can also supply nitrogen, and they've been shown in pot culture to produce some proteinases that degrade protein and release the nitrogen. These mycorrhizae also increase the drought tolerance of the plant. So common desert plants typically are heavily mycorrhizal. I've seen a reference to where cactus are really mycorrhizal. And they can also increase the resistance to root pathogens, even though they don't form a sheath. And I'm not sure if anybody knows the mechanism for that. The growth of the plants that are infected is better, and you can tell, and I don't know what that plant is, but I picked this off the internet. These plants haven't been inoculated with these mycorrhizae, and these have. And you can see there's a definite difference in growth, particularly if the soil is poor in nutrients. If you have a garden where you fertilize and water, then I wouldn't think that you'd want to add these mycorrhizae fungi, because they're not going to do much if there's plenty of water and plenty of nutrients around. But in situations like natural soils, they typically will increase the growth of these particular types of plants. Okay, I was going to stop here too before we go on to the last ones. If anybody has any questions about endomycorrhizae, the AM arbuscular mycorrhizae. Do we have any of those or not? Do I look in chat? I can't see chat.
There's nothing in the chat right now.
Okay, well then I'll go on. How about that?
Wait.
Wait. Okay.
One question from Betty Spence asks, why do they increase drought tolerance?
Because they scavenge the soil for more water. The hyphae are going out into the soil, and consequently you have a bigger volume of soil that's being explored, so that you can take up water more than if you just had the plant roots. And I should have practiced my pronunciation of the term root too, because I don't say that very well, but you know what I mean. Okay, then I'm not going to say much about these last types. Orchidaceous endomycorrhizae. Orchids are kind of interesting because the seeds of orchids are really small, and they don't contain enough organic reserves to allow for the seed to grow up and become a seedling and support itself. So they've got to be infected right after they're germinating, or they're going to die, because they just don't have the type of nutrition in the seed that allows them to grow. So here the fungus is going to provide the seedling with its carbohydrates. If they don't make an attachment to a fungus right after they germinate, then they're going to die. And the basidiomycetes that are involved in these types of mycorrhizae are really good litter decomposing species. And those are a couple of genera, but there are others. And they produce cellulases, and they can decompose materials in the soil to provide the nutrition for orchid seedlings as they're getting established. So the fungi that are involved are widely distributed outside the symbiosis. There are some of them that are plant pathogens. Others are saprotrophs. In other words, they're not pathogens, but they just decompose dead organic matter. And they obtain those kinds of carbohydrates that they then pass to the orchid seedling to allow that orchid seedling to grow without being able to photosynthesize. Apparently, seedlings don't start photosynthesizing for some long time after they germinate. It also appears, since these are some of them are plant pathogens, that there's a delicate balance between the plant and the fungus, and that the orchid's got to keep the fungus in check so it doesn't destroy the orchid, and it digests the intracellular hyphal coils. They may produce antifungal substances, or the fungus will kill the orchid. So it's kind of a tug of war, or a battle between the fungus and the orchid, and one of them has to keep the other in check or die. It's not clear what the benefits to the fungus, but after the orchid starts to photosynthesize, it may obtain various types of sugars from the plant, and it may also obtain various and sundry other kinds of organic chemicals from the orchid. There's a picture of an orchid, I guess. And here are some pictures of cells in which the hyphae of the mycelium, the hyphae of the fungus, has penetrated into the cell. There you can see it better. There's a cell with a ball of hyphae inside of it that is growing in there. And providing the orchid with some types of nutrients. So this is a very different type of a mycorrhizal association than we've talked about with the other two. And it's very specialized. It only occurs in orchids, although there's lots and lots of species of orchids that carry these things out. And then we have a variety of different mycorrhizal types in the Ericales. We have, first of all, the ericoid mycorrhizae, in which the plants are in the Ericaceae. This includes things like heath and blueberry. The fungi here are in the ascomycota, and they form a loose network and then form hyphal coils inside the epidermal cells of the root hairs. And that's where a nutrient exchange is thought to take place. There you can see hyphae coiled up inside the outer cells of the root. These have been shown to supply nitrogen to the plant. Fungi secrete proteinases, and there's not a whole lot known about these, and that's about all I got to say about them. And there's also arbutoid mycorrhizae. Plants are also in the Ericaceae, and this includes things like madrone, which is arbutus, manzanita, and here we are at the manzanita, which we're interested in around here, and wintergreen. Here, the fungi are basidiomycetes that also form ectomycorrhizae. So the fungi will form a sheath and a harding net, like we saw in the ectomycorrhizae, but the hyphae also penetrate into the cells in the cortex. So they're a little bit different than the ectomycorrhizae in that they actually do penetrate into the cortex. And I've never seen the names of the species of basidiomycetes that form these. I'll try to look that up and see if I can find it. And then if you go out there after a rain where manzanita grows, you might find some fruiting bodies of these particular fungi. And if you picked one of those or some of those and brought them back and smashed them around your manzanita, you might be able to get something established there that way as well. That's how I get fungi to grow, is throw them on the ground and stomp on them. Mushrooms. Oh shoot, I can't go. What's wrong? There. Okay. I went about 3. Oh, I can't go back. Oh, there. I went back too far. And then the last one I'm going to mention is the monotropoid mycorrhizae. And here the plants are non-chlorophylous, something like Indian pipe, which is indicated here. People have sometimes called these saprotrophic plants, but really they're mycorrhizal plants. The fungi are basidiomycota, boletes. Everybody know what a bolete is? Bolete are the mushrooms that have pores instead of gills. And they form ectomycorrhizae with other plants like conifers. And so then we have the fungus acting as an intermediary to take organic nutrients from the conifer and pass it to itself, and then pass some of it on to the monotropa, and as well as inorganic nutrients that the fungus takes up. So there's a case where the plant actually parasitizes the fungus, which is getting its stuff from the other plant. So, okay, so mycorrhizae then are key components of ecosystems. I think hopefully we know that by now. And they link plants within a habitat, and particularly in forests. This is sometimes called the wood wide web instead of the world wide web. It's the wood wide web, in which you can take radiolabeled carbon dioxide and feed it to a tree, and then find it in seedlings that are growing nearby. So a seedling that's growing next to a tree or nearby a tree can be connected through its roots and its mycorrhizae to a mother tree, so to speak, and we can have nutrients being passed from the mother tree to the fungus, and from the fungus to the seedling, which allows the seedling to grow in a situation where it might be heavily shaded. And also in ecosystems, these mycorrhizae are going to retain and conserve nutrients, mineral nutrients that would otherwise possibly be leached out of the situation. Okay. Also, it's been found, particularly in forestry, because foresters have been looking at interactions between trees with their mycorrhizae, that certain regulatory chemicals may be passed through the fungus from one tree to another. For example, if if an insect comes and starts to eat on a particular tree, that may signal that tree to start to produce a particular type of compound that will try to offset that damage or to offset the insect biting on it. And that chemical then can be passed through the fungus to other trees in the vicinity so that they may be pre-warned, so to speak, so that they can already start to have a response before the insect actually damages it. And people in forestry have been producing these maps where I think the trees are indicated by these big green circles and the fungi are indicated by their little dots. And you can see the network that occurs in a forest between these different types of trees in the forest, in the wood wide web that occurs. And I might just mention here, if you get Amazon Prime, there's a documentary that Heidi and Don mentioned to me a couple of days ago when we were seeing if I could work my computer, called Intelligent Trees. And it's a very good documentary about the interconnection among trees in the forest. And every now and then they bring up the mycorrhizal fungi. In fact, whenever they do an experiment, they always inoculate. they always make sure they inoculate the soil with the fungi, even though they don't talk about them as much as I would have liked to see them talk about them. So in forestry particularly, they've recognized and used mycorrhizae to inoculate seedlings, particularly if they're going to plant them in reclaimed areas, because it's important to have, if you just take a seedling and you don't have mycorrhizae on its roots and you stick it in a mine spoil with heavy metals, you may go back a little while later and see all your seedlings are dead. So it makes the process of planting these seedlings a lot better if they've already had some kind of a mycorrhizal established on them before they're planted. And that Pisolithus tinctoris has been one of the ones that's really commonly used because it just forms massive amounts of spores that are easily collected and you can grow them. There have been some studies with arbuscular mycorrhizae that have added to crop plants, and they've shown that they have increased growth rates, but these haven't been extensively used. And particularly, you can go out and a lot of places on the internet, you can buy arbuscular mycorrhizae, something that they'll sell you. And I don't know if, I think I'd go out because, all our native grasses are just loaded with arbuscular mycorrhizae. And so if I was going to try to introduce it into a soil that I didn't think had any arbuscular mycorrhizae, I just go out and dig some soil up around some native grasses that are around side oats grama or blue grama, and add that soil to the soil that I had. And I think that you get some good arbuscular mycorrhizae, but you'll never know unless you want to get a microscope. And now, one way you can detect arbuscular mycorrhizae is you can sieve the soil, because these spores are big enough. If you have a fine on a sieve, you can collect them on the sieve. And then with a dissecting microscope, you can count how many spores there are per 100 grams of soil or something like that. But you have to have the specialized sieves to do that, and a dissecting microscope. So it's just really hard to determine whether you have these things or not. But you can, if you really want them, I'd go out and pick up some soil around some grass and put it wherever you want it. Okay, I think that does it for me. If you got any other questions, I'd be happy to try to answer them.
Okay, so Keller.
You're probably muted. Are you muted? Okay.
Keller.
Yeah.
So Russ has a question that says orchids have thick coral-like roots, in parentheses. Are they ectomycorrhizal, an exception to the rule that ectos are usually on trees?
I don't think so. I think they have mycorrhizal, the saprotrophic or pathogenic mycorrhizal on orchids. I'm not an expert on orchids, but I think that's the only time I've ever seen mycorrhizae associated with orchids. They're the things like armillaria or other basidiomycetes that are really good decomposers, and they may form thick thickenings on the roots.
So another question.
And if Russ is there, I don't know what kind of mosses form mycorrhizae either. Because I did see that some mosses will form mycorrhizae.
Russ, would you like to chime in?
No.
Okay, so Ken has a question.
Okay.
Ken says that no-till vegetable gardening is thought to be beneficial because it does not disturb the mycorrhizae.
Mycorrhizae.
What do you think about that? And would addition of these mycorrhizae be beneficial to the disturbed soils?
Would mycorrhizae be beneficial to disturbed soils?
Well, what he's saying is that this no-till or very minimal till vegetable gardening, where you don't use a big rototiller and dig down deeply into the soil, would that be beneficial?
The mycorrhizae.
In other words, I think what he's asking is that would that be less apt to disturb those mycorrhizal relationships? And if so, maybe could something be added? Could the mycorrhizae be added to the disturbed soils?
Yeah, if you're gonna rototill the soil, then if you wanted to add the mycorrhizae, like you could add spores that way to the soil. I mean, once the plants have died, the mycorrhizae aren't going to be living. They're just going to be as spores in the soil. So I don't think that would make any difference at all to the mycorrhizae. But if you've got a garden and you water it and you fertilize it, then mycorrhizae aren't going to be real important anyway. That's the whole thing. If the plants have enough nutrients and they have enough water, then they're not going to grow better with mycorrhizae, I don't think. They're only in natural environments is where you're going to see the big difference.
One of the things that I'm thinking about organic gardening and organic vegetables, crops, etc. And one of the things that I've been thinking about is what organic gardeners and organic farmers are doing, whether the products that they produce are better for us or not. One of the things that they really try to do is they care for that soil. And what in my way of thinking is that care for the soil is an extremely important part of farming or gardening. And I think maybe that's kind of what Ken maybe was getting to a little bit. I mean, it's time that maybe we stop treating soil like dirt.
Yeah, except if you add compost and other types of organic materials to the soil, that's going to enrich the soil as well, because then when that decomposes, that releases nutrients that the plants can use. You just don't want to add a lot of like straw, which is very high in carbon and low in nitrogen and phosphorus, to a garden right off the bat. You want to let it decompose a little bit so that the nitrogen and phosphorus concentration increases, such that then it would be available for the plants and not just for the fungi that are decomposing the straw or whatever other organic material you put in there.
So Suzanne has a question. Can these systems be utilized in phytoremediation? How do they deconstruct the contaminants?
Oh, you mean the heavy metals and things like that?
Suzanne would like to
Suzanne,
unmute.
if you'd like to unmute. And
yes, what do they do with the contaminants? Do they break them down into?
No, they sequester them probably, and they don't let the plants get into the plant. The fungus would sequester something like mercury or something like that, and the plant wouldn't get it.
So, but if the fungus sequesters the mercury is still there.
Yeah,
So-
oh yeah, no, they can't get rid of it. There's no way they can get rid of it.
Okay, so they can't get, because there are other plants that can deconstruct. They take it up and then you just take the plant away, and you have to like treat it like a hazardous material, you
right,
know.
right. Yeah, no, this wouldn't do that. This just doesn't. The plant is more tolerant if there's heavy metals or something like that in the soil. The plant doesn't get it. The fungus gets it first, and then, so the plant's not affected.
And so, but the fungus does not deconstruct it in any way.
No,
Still, okay,
no, yeah.
still there.
The plant doesn't deconstruct it either. You take the plant away.
Right, you take the plant away, right,
And you can't take the fungus away because it's microscopic.
right.
So,
Maybe that's not such a good idea then.
So Keller, so Betty has a question. Does the idea of interaction of mycorrhizae between trees throw doubt on the virtue of thinning forests?
there's that documentary that you gave me has a very good discussion of those kinds of things. And I don't know as I'm qualified to go into that in too much depth, but yeah, you have a problem if you thin too much. And if you clear cut, just plant one species back, you have a problem because you don't have the same type of interaction with different mycorrhizae and different trees that you would have in a natural forest that is composed of more than one species. So yeah, I mean, there's a problem there. And also taking down big trees. There's a German, you get a little bit of German in this documentary too, because if you listen to the guy, there's one German forester who likes big beech trees. And you don't want to get rid of all the big trees because they kind of act as a memory for all the little trees that are sucking out their knowledge. This is a little weird, but that's the way they think about trees if you're a forester.
So Betty and others, we watched and the video that Keller refers to was on Prime, and it was Intelligent Trees. So if you're interested in watching that.
Yeah, that would give you a really
Thanks.
good idea. If you can't get Amazon Prime, you can come over and I'll let you watch it.
There we go.
Once COVID is over.
And bring pop.
Intelligent trees, okay, thanks.
Intelligent trees.
So Mary has a question. Does a mulch of wood chips promote mycorrhizal growth that might be already present in the soil? What
No.
else promotes mycorrhizal growth?
The tree
The tree.
or the plant. That's what promotes mycorrhizal growth. Yeah, because they're not going to be decomposing the wood chips, so they're not. Now, wood chips may stimulate fungi that grow in the soil and form mushrooms and other things, but not the majority of the mushrooms. But lots of fungi will decompose wood, so that can make more mulch but it's not going to affect the mycorrhizal fungi, except maybe to retain moisture.
Yeah, if I could interject here.
Yes.
Foresters,
30 years ago, I was involved in a project where we were planting a new boulevard with 2 species of oak trees. And that was when we were first able to buy mycorrhizal fungi. And we treated about half the trees with the mycorrhiza. The other ones we didn't. Now these were 5-inch diameter trees, so they were good size, coming from a nursery, and all transplanted with the big tree spades. And we expected to see some improvement on the ones because this was construction loam, as I called it, anything that the contractor had dumped in the middle of the boulevard to meet his obligations, and it was terrible soil. But anyway, we did not see any improvement at all with the ones where.
What kind of mycorrhizae were they? Endomycorrhizae?
Yeah, it was ecto. I mean, these were oak trees.
Yeah, but what did they give you? Mycelium or
Pardon?
what did they give you? Mycelium or pellets or something or
It was a liquid.
liquid?
Yeah. Anyway, but that was a long time ago, and
Yeah.
we didn't get any results out of it. And then I was talking to folks up at Morton Arboretum that just does a lot of research on trees. And they said, well, the trees probably had plenty of mycorrhiza on their roots, so it didn't make much difference at all.
Yeah, that could be.
Yeah.
Or it could be that these people that sold you this liquid didn't have anything in it.
Very possible. But anyway,
Well,
it didn't work for us at that time. But that's been a long time ago, too.
Right.
So Keller, there are no more questions on the chat.
Okay.
So I guess some takeaways here.
Betsy's hand is up.
Think about the fungi on the
Betsy,
roof when you go
Betsy's
out.
got her hand up. Go ahead and unmute, Betsy.
My hand was applause.
Oh,
Okay.
Thank
sorry.
you, Keller.
Okay. Somewhere you need to find two hands.
Russ has
Oh,
it.
2 hands? Okay.
Okay, see, I think Pamela has the waving thing and thumbs up. Russ, do you have a question?
Yeah, let me rephrase that question I asked on chat a little bit. If the few times I've actually dug up an orchid, they really have thickened. It's not really roots even. It's almost like, it looks like you're digging up a coral.
Oh.
So you said that was, huh?
Maybe it is ectomycorrhizae if it looks like coral.
Yeah, it looks, in fact, they even call it a coralloid rhizome is what they call it.
Oh.
It's thickened. It doesn't look like roots at all. And I
Yeah.
was wondering, that's why I wondered if that might be ecto on a small plant.
Yeah, whatever I've read, it's not, they're mainly just woody, ectomycorrhizae are on woody. If you can say, if you get one time, section it and see if you can see a Harding net, then you'll know.
I might get you to help me with that.
Okay,
Thank you.
sure.
Any other questions?
I have a comment going
Yes.
back to Betty's question about tree thinning. I was a volunteer wildland firefighter for 4 years, and during that time, I really learned the importance of thinning. So I would think that anything you might lose with mycorrhizae is way balanced on by having fewer trees, but as Keller said, it depends on how many trees. We've had our 30 acres thinned. A third of the trees were taken out, and the ones that remained are doing very well. And I feel much better about when, and not if, but when the wildfire comes through where I live, I think we'll be in much better shape.
Yeah, and I guess Betsy and the rest of you, that's the importance of research. And, as time goes on, we hope we get closer and closer to the way things really are and what is really the way to do things. And so we really need to support our scientists. And I guess another takeaway would be that we should stop treating soil like dirt, like I said before, and that Keller is a really fun guy.